Ilse?
Yes.
Kan ek gaan nou vir die presentation mode, is daai reg?
Yes, perfect.
Ek kon weer werk.
Okay. Okay, I think we can start. Thank you very much for that, Francois. Thank you everyone for joining tonight. We are privileged to have not one, but two very fantastic speakers tonight on the topic. I just wanted to, before we start, say a thank you to Pfizer for sponsoring this event, and they in no way had any influence on the content of the talk or the topics of the talk, but thank you for the financial support. As I said, we have two speakers tonight, so I'm going to give you both of their bios as an introduction, and then they will, between them, just switch between their screens and who will be speaking. Just another, sorry, there I lost my screen. Another thing to keep in mind is we're not going to take questions before the first speaker session. We're just going to take questions after both of these sessions because they are integrated talk. If you want to leave a question, you're welcome to use the Q&A function at the bottom of your screen, or you can type in the chat box. We will then repeat the question and get you some answers. Okay, first bio tonight, the first speaker, we have Professor Francois van der Westhuizen. He's a professor in biochemistry and currently also the Deputy Dean of Research and Innovation in the Faculty of Natural and Agricultural Sciences at the Northwest University. He has been a researcher for almost 25 years on mitochondrial metabolism and mitochondrial disease etiology in South Africa, with a recent expansion to neuromuscular disease. In this regard, he coordinated the participation of South African researchers in the ICGNMD study, and thus will provide the introduction and the overview of the study. We're also privileged to have Dr. Maryke Schoonen as a speaker tonight. She's an early career research fellow at the Center for Human Metabolomics at the Northwest University. Dr. Maryke Schoonen earned a PhD from the Northwest University in 2019. Her postdoc research has been dedicated to studying rare diseases with particular emphasis on neuromuscular disorders. She's actively involved in the ICGNMD consortium. As mentioned earlier, Francois is the person who established this connection for South Africa, and ICGNMD stands for the International Center for Genomic Medicine in Neuromuscular Diseases. Dr. Schoonen received specialized training in variant interpretation and evaluation from the ICGNMD at the University College London, enhancing her expertise in this critical area. As an early career researcher, her primary focus is on congenital myopathies in South Africa, striving to advance the understanding and treatment of these conditions. Thank you both very much for dedicating your time tonight. Maryke asked that I mention the number of attendees. So currently we are about 70, exactly 70 attendees in this call, and people are still joining. There, just one more person joined again. Thank you everyone for joining again. And yes, I'm going to give over to you then, Francois and Maryke. Thank you very much.
Thank you to give this talk. Can you hear me, Ilse?
Yes.
Audible? Yeah, thank you.
No.
Yes, so the reason why I'm introducing actually this study is even though this presentation tonight will end up with case reports, which is the main aim of the talk, this is a research study that was initiated from which there are a lot of potential that I'll explain. And one of these outcomes are indeed, individual case reports and outcomes for diagnosis. But I have to reiterate from the start that this was a research, or this still is a research study, and there's a lot of people involved in this, which I'll give an overview for. And I think once Maryke starts to present some, at least her work there, you'll understand better the background from where the data comes from. So I'm sure you're probably familiar with neuromuscular diseases, which is quite a highly prevalent rare disease affecting around 1 in 1,000 individuals. It includes many different disorders of muscle and nerve function, and about 20% of all neurological diseases is neuromuscular disease. Most of these are single gene disorders, and there are many different phenotypes that can be classified genetically. Very important for us, which we know from before we started with the study, is there for many neuromuscular diseases as a group, a genetic diagnosis can lead to effective treatment. And obviously, understanding therefore the etiology of neuromuscular diseases in the population varies greatly and will improve the diagnosis and the treatment. So I want to reiterate also the importance of the population that we are working in, and the result, or to illustrate this, around about the time where we started with this study, and we were primarily mitochondrial disease research group, in our NHLS genetic screening or testing for neuromuscular diseases as a whole were very, very limited. And this is a table that illustrates the number of phenotypes and the genes that were tested at around about 2017 when we motivated this study. And you can see here that it's very limited in terms of mitochondrial disease. It was probably a bit better, but for the other diseases, it was very limited. And in addition to that, the screening or next generation sequencing of patients really happened in an ad hoc basis, as far as I know. And many of those were private, and those results were never reported for us to be able to determine what are the causes for these diseases in our populations. So from the collaborations that we had in the UK at that time, we were aware, and they were aware of us, our involvement also at the time. But at that time, around about 2008, 2018, there were I think it's 3 universities that formed the MRC Center for Neuromuscular Diseases in the UK. And their objective, they were obviously UK-based, very specifically UK, not out of the boundaries of the UK. UK MRC funded. Their objectives were cohort building with of about, I think it was about 1,000 or so patients that they were recruiting. And the aim was experimental medicine trials, genetics and biomarker studies, also biobank development to collate and accumulate material, gene discovery, and translational research, education, training, and networking. But at the end of this study, which was the preamble to our study, was that they realized and they had to move towards a more global, more population diverse, for their own sake, a more diverse population study. And this is where they motivated and recruited their collaborators across many countries and about 4 continents to form a global network, which was called then the International Center for Genomic Medicine in Neuromuscular Disease Study. So this is a research study, I emphasize that again. And this was initiated, or this started in 2019 and ended in May this year. And this study is a global 4-continent study between the UK, Turkey, India, South Africa, Zambia, and Brazil. And later on, the Netherlands joined in a technical capacity as well. And at each of these countries, there were a couple of centers. I'll come back to that later. So the purpose of this study, I'm going to diversify the objectives here. So the first was to create a transcontinental genomics research and capacity building partnership between the UK and these LIMIC countries. There were 5 of these countries at the start. So again, the objective was to expand the current UK cohort to include more diverse populations and to aim to get to around 10,000 patients, of which South Africa, we aimed that we would be able to get to about 1,400 participants, not only probans, but families included here. And to have these participants have an accurate genetic diagnosis. That was a major aim for this. And you'll notice also trial-ready cohorts. So it means that these cohorts had to be very well defined phenotypically and genotypically to be able to have cohorts of this kind. So, and along with that, all the other opportunities that cohorts will present. The third objective was to discover new disease genes, of which we were very excited of in our understudied populations, and to understand the comparative genetic architecture and disease mechanisms, which is mostly focused on that second phase that we aim to achieve. But then again, also human capacity development is a really key objective of this study. So there were academic, mostly clinical partnerships where clinical fellows were assigned to each of the countries. South Africa received 2 from this agreement, with a training program to generate clinicians and clinical academic researchers, really, that could sort of come into to follow up on the current research that we currently are having. The fifth objective were to establish international bioinformatics pipeline and to translate these platforms to the various countries, also partly to train this and to help to develop sort of pipelines that can be used in the diagnostic setting also in these countries. And then finally, to translate these to, as I said already, actually to diagnostic opportunities and to improve the research capacities and networking that was needed. So essentially, and Maryke will again give a much more interesting workflow. This is the workflow that was finally adopted after many changes. This was what was done in the end. So you can see here the recruitment is always the first phase, followed by, I'm not sure if you can see my cursor here, but followed by a deep phenotyping process of the clinicians and the fellows, sample collection, and those samples either were transferred to the UK for sequencing or locally sequencing. We had options to either sequence them locally or to have them transferred to a service provider that was coordinated by the UK. Then this followed international review of the data with the fellows primarily involved and the bioinformatics team there. Then there's a genetic testing this is actually the decision of what to test for was first made before the genetic tests. The genetic tests include SNP analyses, local analyses, screening, but also proband, whole exome, whole genome sequencing, and family whole exome or whole genome sequencing. And then there was a data analysis part that I don't need to talk to you about too much, which in the end resulted in an outcome of either solved or unsolved. And then obviously, as this is a research study and we want to disseminate the data, we are still in that process, obviously, to publish, to collaborate further, to develop the data further, and then to archive and share data in the end. So this is essentially the workflow, and as I said before, Maryke will again talk to that. So I'm going to just briefly tell you what the results were. So there was one publication near the end of the study that came out. So this is maybe a year and a half old, this publication, but nevertheless, you can get an idea of what is the outcome of the networking. And important here, I share this image just to show you there were about 50 people involved in this. And there were probably much more, but these were the authors of this publication, original contributors to the initial data. So in the end, there were about 100 researchers, 18 centers, and over 7 countries that formed this transcontinental genomics partnership. Also from this publication, it was presented, the trial-ready cohorts with their proper genetic and phenotypic diagnosis were presented in this publication. This publication showed the first 6,000 recruits for the study across all the countries. You can clearly see the larger countries like India and Brazil had quite a lot of patients or recruits for this study, and you can also see the breakdown of the phenotypes that resulted from at that time. You can see limb-girdle muscular dystrophy is quite a great number, and in India, for example, you can see the remarkable number of patients there. We know that congenital myopathies are quite significant in South Africa, and you also see that presented in this data. But this is just an illustration of the cohort building that was achieved and is still continuing at this time. So important outcome of this at that time was to show that, and even at that time, that all the genetic data weren't available, especially the whole genome data. But at that time, we could already see that 56% of the probands were solved or possibly solved as a result of this study. And quite interesting also and important for us, which we probably expected as well, is that about 1 in 3 of the variants were disease-causing variants were considered as novel. Now, just in terms of gene discovery and understanding the genetic architecture, here is an example of what was found, and this compares some patients across a number of the countries, and the relative distribution of the splice sites and deletions, duplications and nonsense variants that we found between South Africa, India, and the total cohort of Duchenne's muscular dystrophy patients that was recruited. And you can clearly see a variable distribution of these different types of genetic causes across these countries, with South Africa having much more nonsense mutations or variants, single variants, that compared to deletions, for example, that's much well known for this disease. Right, so if we look at South Africa and we want to get closer to the cases that Maryke will present, so the notable outcomes for South Africa thus far, and we are not finished yet, although the recruitment has finished, the results are not completed yet. It will take some time, obviously. Just a brief overview of how this study was sort of developed at the time. So important to note that when we started the study, we received, we basically received 2 fellows from which formed the core of this study. And one fellow was adult pediatric, or sort of adult neuromuscular fellow, and the other one was a pediatric fellow. So the adult fellow was shared between Stellenbosch and University of Cape Town, and the pediatric fellow was shared between University of Cape Town and the University of Pretoria. So we had to do with these limitations in terms of capacity. There was not much funding in terms of logistic support. We had to generate some of that funds or get some of those funds ourselves. But there was some limited funding that was provided that could help us logistically here. And the idea was that we had these 4, actually, if you count Northwest University, and I'll come just now to what each site does, we were basically 5 recruitment centers in the end. And those centers, the idea was that they collaborate with close centers or centers close to them. So you'll see here that there were 2 pediatric centers at University of Pretoria and University of Cape Town, and 2 adult centers, both in the Cape, Stellenbosch University and University of Cape Town, at Tygerberg and Groote Schuur. And then Northwest University, where we're situated, we had, although we are a diagnostic center, we did not do much of recruitment except for some of the recruitments of metabolic myopathies. But we are a research center, and we assisted more in the northern provinces, especially University of Pretoria. So these are the participants at this time, or as they developed from the start. Now, in terms of the cohorts, you can notice here again the 4 centers here. And we, as I said before, we aim to recruit 1,400 participants, of which at the time that we presented this data, it was earlier this year, we had about 1,135, of which 54% were probands. And obviously, some of these probands already had a genetic outcome, but we sent many of them still to have whole exome or whole genome sequencing. I must admit this data is a bit outdated. We don't have access to all the data at this time, but this is, I know the number in University of Pretoria is over 600 at this time. So at the end, the final number we'll get once the outcomes have been sorted out. But you can see here the solve rate were about 64%, and I have to admit also that some of these patients were already diagnosed locally. So the demographics of these patients, 45% were Black African patients, 2% Indian, mixed ancestry 20%, and white 33%. Right, in terms of the disease distribution, between these 4 centers, you can also see here for many of these centers, obviously they have different focuses, they have different expertise and capacities. So they did not have obviously the same recruitment outcome of the different disease types. For example, you can see in terms of the most frequently recruited one, or the highest number of recruits for congenital muscular dystrophy and myopathies, you can see Pretoria and Red Cross, which are both pediatric centers, had more or less the same number of recruits. And even with that, there were differences in the outcomes there in terms of the phenotype and genotype. Then you can see for the rest, depending on whether it's pediatric or adults, you'll see a differentiation between the types of phenotypically recruitment. And again, I say that it also depends on the expertise and the capacities of these centers. Right. So in terms of outcomes, and I'm only going to report here or summarize to you what has been published thus far. There are other outcomes that was not published yet, and so I will leave that out because that needs to be peer reviewed, obviously. But you can see here already. This is a summary of some of the outcomes. I already mentioned that initial outcome about the variable genotype phenotype in not only African patients but other countries or other population groups. And we are knowledgeable about the pediatric outcomes mostly, and there is a very clear variability between the phenotypes and the genotypes of the population in Africa compared to the European data that we had before we started with the study. One specific study that we completed and published earlier this year was a South African multiple acyl-CoA dehydrogenase deficiency, or also glutaric aciduria 2. We built a cohort over many years before even the study started, but we revisited those patients, built a cohort of about 14 patients, 12 families, had a very detailed clinical, biochemical, and genetic spectrum analysis, and we were able to determine that there are 2 variants that need to be screened for, and we could differentiate between the outcomes of these variants. So that's a publication from Michelle Bishop, which also completed a PhD on that. Then from the Cape University of Cape, Cape Town, there were a mutational profile of the SA cohort of inherited neuropathies and spastic paraplegia that was completed, published. There was also an outcome of mitochondrial DNA disorders in these patients, compared also with the different populations that was studied in this study. And then recently, and this is probably appropriate to look at these case reports, there was a patient with a novel PEX11B variant that had a different phenotype than those published already. A very rare disease that Frans Lou Henning and his group recently published, or is still in press. So these are some of the outcomes. Then very important, as I say, immune capacity building is very important. It should not be underestimated. Not only the fellows, but also many people around them. Also, I mean, the PIs, I mean, we all benefited from the study in terms of training and building capacity and new knowledge. So there were a couple of PhDs that was either finished or will finish this year, and they are listed there. But there are also some other students that's also involved with this, and especially the follow-up studies and postdocs that were that could to link up on these cohort studies. So, and then also, we benefited a lot from the bioinformatics exposure, and the development of pipelines We had a workshop, for example, at some time 2023, where we could also, translate this a bit to the NHL's lab people that we are collaborating with. So this is what was quite beneficial for us to be involved with. So the question is, I'm sure you can also ask this. Obviously, we're going to talk about, there will be case reports, etc., that will be discussed during these meetings, and some of them will come from the study. But in general, it's very fair to ask. So what was this? What is the benefit of this? So if we first of all look at the patients, I think there was about at least 300 patients that got in South Africa that got a genetic outcome from this study, and there are many more that we are still waiting for. Unfortunately, whole genome sequencing is taking much longer than we hoped for. But there are definitely genetic outcome for these patients that receive clinical diagnostic reports and genetic outcomes from the study as a result. And hopefully then from that also treatment strategies for that, and having answers to these families who previously were not able to get answers. So even though you can say that this study could have been done in South Africa locally, this certainly fast-tracked things for us in terms of patient outcome and diagnosis. Yeah, I can maybe also add that for patients, there's always the prospect, and that we hope for, is that once you have cohorts, and there could be trial-ready cohorts, and there's opportunities for therapeutic testing or clinical trials, that we have these cohorts available and ready and able to contact them to potentially participate in such clinical trials. But for the clinicians, scientists in South Africa like us, the value obviously is to have better knowledge of the etiology of neuromuscular diseases in a more expanded way, in a more systematic way than before. I'm sure it's much better than the knowledge that we had when we started with the study, and it certainly would increase also other centers that will start to collaborate or already collaborating with this. The hope is that there will be a contribution to clinicians for their ability or the capacity in South Africa to provide answers to clinicians and patients. For scientists, obviously, there's a lot of discovery to be done in our populations. We saw that from this study, but we knew that before that as well. And there is indeed an opportunity to contribute to very relevant knowledge, and even for patients in these cohorts that can undergo early screening for treatment that can be effective. So these are just a couple of ideas that I can briefly think of now. You probably can recognize more than that already. And then for what's next? So we are indeed, the cohort building was done. There are a lot of undetermined cases, and there are a lot of patients that do not have answers yet, and these will result obviously in more scrutinizing of the data that will still come, that needs to be followed up and verified and provide answer to patients. So that's definitely part of what's next. But also what is part of what's next is that from all these data, we barely scratched the top of the iceberg, or we really just picked the low-hanging fruit at this time. There are so many opportunities still to be explored in terms of functional studies that need to be done, of values of unknown significance, even further genetic tests if it's required from answers that we do not have yet. Even after whole genome sequencing, it's likely that there will be at least 30% of the outcome not known. So indeed, so there's opportunities for future research and future collaborations of this. And as Maryke will show you, there are still a lot of things to be explored, and she will now present after this concluding slide of mine, a specific case that she will present as an example of what which is not cohort building, but is individual cases that hopefully can be resolved. And I think I'm going to stop here and then going to ask Maryke. I'm going to stop sharing and then Maryke can take over. I hope my mic was open, Ilse.
Yes, no, we could
Okay.
hear you very well. I just
That's
think
good.
it's that you can't see or hear the audience. They're very quiet.
No.
So it seems like you're alone, but thank you.
Okay. No, I was just making sure. So just kidding. Maryke, over to you then. Thank you.
Thank you. I assume you can see my screen there for the presentation. First of all, thank you for the opportunity to talk about one of our patients that was the longest in our, well, she was part of my PhD cohort already, so we walked quite a long road with her, and I think she's the quintessential definition of diagnostic odyssey. She unfortunately, or not unfortunately, but she passed away earlier this year. And she also always wanted her story to be told. So I'm going to tell her story. And I hope you can appreciate that she not only wanted the story to be told, but she also wanted people to, or other patients to be helped in the future if they have a similar problem, similar disease. So eventually she was diagnosed with Maryke.
Maryke,
Yeah.
sorry, we can't see your screen. I'm not sure if you are showing your screen already.
Sharing. Okay, wait, let me just double check this. Just hold on. Where is it now? How do I get into full mode again?
Are you sharing the correct screen? We can just see your camera.
Yes, share. Is it
There
sharing?
you go. Yes,
okay,
perfect.
Okay,
Sorry for that.
Yeah, so now you can see actually the actual screen. Okay, so is it still sharing? Yes.
Yes, perfect.
Okay, yeah, so she was eventually diagnosed with Maryke. I hope I'm saying it correctly, muscular dystrophy, and I'm going to give some feedback on that. Just, again, the complexity of neuromuscular diseases. So there's not just one. There is a great deal of neuromuscular diseases. And to make it even more complicated is that there are so many phenotypical overlap, which makes the complexity of the clinical diagnosis even more difficult. And I'm sure a lot of clinicians in the audience will agree with me that it's not as easy as just, seeing a patient and immediately know this patient has a Duchenne's muscular dystrophy or Friedreich's ataxia or whatever the case may be. So it is quite a challenge to diagnose neuromuscular diseases. And if I can just show or explain why I say that. So first of all, the timing and the age of onset differs. Sometimes it's in the neonatal period, sometimes it's in infancy, sometimes it's even in adulthood. And the disease penetrance and expression is also an important aspect to keep in mind when you're looking at the genetics. It is so super heterogeneous. Not only the phenotypical side, but also the genotypical side is very heterogeneous. As Prof. Franchal mentioned, sometimes it's one gene that causes a problem, but there are so many genes, and they each have their own phenotypic outcome on a patient. So it's quite complex to diagnose a patient, and that's why they often say we have a diagnostic odyssey in a patient. And I hope we can continue with our diagnostic odyssey in the future, and not just for this one patient, but for the many others that aren't diagnosed as yet. So just to talk about the muscular dystrophies, you will see there's quite a spectrum on which they lie. So at the very top, you have your congenital muscular dystrophies, and at the very bottom, you have your congenital myopathies. So the big distinction between these two is the CK values in the muscle. So congenital dystrophies, muscular dystrophies, usually have an elevated to really high CK level. and congenital myopathies have a slight elevated CK level. So that's already one distinction that clinicians can make when they try to diagnose a patient with a myopathy or a dystrophy. And then you'll see there's a great many deal of types of dystrophies. You have the limb-girdle on the one side, and then we are going to focus on the Emery-Dreifuss muscular dystrophy. That's right over there. All right, so exactly what is this EDMD that I'm going to refer to from here on? So it's a neuromuscular disease that's characterized by muscular weakness and atrophy in the muscles with early joint contractures and cardiomyopathy. So the first three, yes, is it three or is it four? The first 3 or 4 cases in South Africa was reported back in 1987 already with EDMD, and they, as far as I know, were the first South Africans to be diagnosed, first South Africans to be diagnosed with this, and I haven't seen any more publications, recent publications, but I'm sure there are a lot, as there is about 61 publications on the variant that we found alone. So the inheritance pattern is, there could be two ways about this. There's an autosomal dominant one with the alanine A gene that's involved, and also an X-linked one with the EDM gene that's involved. So the age of onset is in childhood, and the prevalence, this is a global prevalence. Unfortunately, I couldn't find a South African prevalence, but the estimated prevalence globally is 1 in 400,000. So it's a really rare type of muscular dystrophy, but nonetheless important to identify. So basically, it's a clinical triad that is comprised of joint contractures, elbow and posterior neck tendon contractures, and obviously this worsens in childhood as everything progresses with the muscle weakness going forward. So to come back to Prof. Francois' diagram that we followed in the ICGNMD process, is all of our patients in the northern parts of South Africa was recruited at University of Pretoria at Steve Biko Hospital. That was our referral area, and we did some really deep phenotyping and clinical details. We collected that after obviously the assent and consent and all the ethical procedures, and then we collected samples from each of the patients. What I did not show here is that the sample collection was first stored at Northwest University, where we did DNA isolation before we sent it to UCL, where they performed the genetic testing on our behalf. They sent it to the Netherlands, as Prof. Francois mentioned. They became a testing site later on. So for DMDs in South Africa, you can test it locally for at the NHLS, this and for the SMA gene, the SMN1 and the DMD gene. If that is negative and we can't find an answer locally, then the children were or the participants were included then for the whole exome sequencing and later on whole genome sequencing. That was first the proband was sequenced, and then the if an answer wasn't found in the proband, then the whole family was considered to be sequenced. If we did find something in whole exome sequencing that looked like something that could be the cause of disease, we did segregation analysis, firstly to identify if it's the disease inheritance pattern, and also to confirm the variant. It's very important. That variant should be confirmed before a report is given out or any further biochemical tests is being done, or in vivo and in vitro tests are being done. So it's very important to do the golden standard of Sanger sequencing, and that was done at our university. And we did this for all the variants that we identified that's possibly disease causing. So when we find a variant and the Sanger agrees and everything, all the ICGNMD criteria agrees with, we could then give feedback to the patient and consider the patient as solved. So I'm going to explain basically using the number system here for my patient, and just so you can follow the process that we had to give her an answer and her family an answer. Okay, a bit on her, bit of her clinical. I don't expect you to understand everything. It's just a broad overview of all the symptoms she had, the HBO positive terms, and obviously HBO positive terms are just a look into the whole picture. It's not the whole picture, as Prof. Chris always said. HBO terms is like a puddle of water, and you can't imagine the iceberg it was before. You don't know the structure of the iceberg. It's just melted water at this time. So it's important to have HBO terms because it's a standardized term, but it doesn't tell the whole story. I'm going to show you why I say that just now. The important thing I want you to notice is that her CK levels were extremely high. She had muscle biopsy, and from that muscle biopsy, we also did mitochondrial analysis because that's also my background. And we saw that she had a complex 1 and complex 4 deficiency. So initially we thought it might be a mitochondrial disease, but her symptoms and signs, it didn't quite agree with mitochondrial criteria to be a primary mitochondrial disease. So it was something that we always wondered about. The other important thing is her DMD test was negative. So she did show signs of a muscular dystrophy, but not DMD. So that was also an interesting part. This is our patient. Just to show you the level of severity that she had to live with. She had, if I can just show you as the phenotypes pop up, she had extremely difficulty in walking. She had to assist herself with her hand, as you can see on her hip over there. Her neck was really weak. She couldn't keep her neck in a normal position. When she had to eat, she had to lay down and eat so that the food can move in a horizontal direction so that she can eat without discomfort and anything like that. So she had all the symptoms of a typical muscular dystrophy. There was just, we did not know which one she had at this stage. And then the ICGNMD study came, and we were able to recruit her at the age of 15 years. And she showed her first signs at 7 years of age. And I think she was first in our clinic at the age of 11 years, if I'm not mistaken. But yeah, she was recruited at 15 years and 11 months. Oh, and also very important, she had sick sinus syndrome, and eventually she passed away due to heart and cardiac complications. So the genetic findings that we identified is a very well-known pathogenic variant in the alamin A gene. It is a heterozygous one, and we also confirmed that it's de novo. And it was, I will show you in the next slide why we say so, but according to the ACMG classification, it is definitely pathogenic. As I said, there was more than 61 publications on this gene alone. The African allele frequency I was able to see, and that was really, really almost non-existent, and no homozygotes ever reported. So that kind of gives us an idea of the severity of alamin A, this missense mutation. So this is just the at the bottom there, you will see the gene structure and our variants situated there at the very end of exon 7. With our segregation analysis, we confirmed the heterozygous mutation. As you can see over there, I'm not sure how to use my cursor now, but you can see with the bright markers there, it's the heterozygous. The mom and dad were wild type for the variant, and basically her family pedigree also showed that her other sibling was not affected at all. And as I mentioned, this is a well-established variant, and it was first reported by Bonnet et al. In 1999 already, demonstrating a arginine to tryptophan substitution. Why is it not moving? Sorry, I'm not sure what's happening now. There we go. Yes. Okay, just to give some background on the pathophysiology of this. So in the muscle cell, you have your myofibril structure, and you have your sarcomeres in the muscle cell, And when you enlarge this sarcomere, you will see there's a lot of things happening in the cell. So just a disclaimer, this is super, super simplified. This is really not a very detailed illustration of what's really happening. I'm just trying to make it as simple as possible. But basically, you have your calcium channels where the action potential activates calcium release from the ryanodine and the SAC3 proteins. And then you have your sarcoplasmic reticulum, which the mitochondria is quite closely involved with. Mitochondria is actually part of the whole cell. It's not just near that part. You have your actin filaments that keeps the cell intact, of course, and then our lamins A that encodes the albumin A gene is right here in the nucleus. So the main purpose of the lamins AC is to keep the integrity of the cell. And once there's a mutation in that gene, everything goes haywire, the integrity gets lost, basically your whole muscle cell it doesn't know what to do. It's all over the place. So the outcome of that is that when your plectin gene binds to your sarcomere and your intermediate filaments, they can't do that properly because plectin is one of the key chains that's between your intermediate filaments and your lamins. So if the lamins are broken, the nucleus integrity gets lost, the plectin mechanism gets broken, and everything in the cell goes haywire. Calcium can't get released, mitochondria is affected, and that's why we suspect she had a secondary mitochondrial defect, because of everything going haywire in the cell, the mitochondria is also affected. And that's why she had the mitochondrial defect. And it's actually a brilliant kind of nice situation, nice story for us to clear that up as well. So as you can see here, it's the lamins are required for the mechanical integrity, as I said, and if something were to happen, it can cause some really damaging effects on the muscle for muscle weakness and atrophy and everything that's associated. So going forward, and the take-home message that I want to leave with you is that we now know what to look for in patients with her type of clinical phenotype in immunodeficiency muscular dystrophy. So early and accurate diagnosis is crucial. You can distinguish it clinically, metabolically, and genomically between different muscular dystrophies. I'm not a clinician, so I can't exactly tell you which specific phenotypes is in which patient, but I do know that Duchenne's muscular dystrophy, the children are easily bound to wheelchairs, whereas our EMDM patients, they could walk still up. Our patient could walk until the day she died. So it's the early onset is not as common in EMD as in DMD, but there's also a very big difference there. So if you know your patient and you know your muscular dystrophies, it's always good to, if the DMD is negative from our local testing, to look wider. And with that in mind, we like to encourage collaboration between the clinicians, geneticists, researchers to facilitate accurate diagnostic. And this is obviously a multidisciplinary approach. And for the treatments going forward for each patient, it's quite important. As I said, we know now what to look for, and we can facilitate some treatment on our other patients with the same genetic defects to help them going forward, because some of them are still quite young, some of them are also beyond treatment. It's just symptoms management. So yeah, and then I would like to also name that we do have support and advocacy communities such as Rare Disease South Africa that are there for patients and family to support them when they are finding themselves in this difficult position of having a rare disease, because it's not only the patient affected, it's the whole family that eventually is affected. And with that, I say thank you, and I hope you learned something from our EMDM patient. And yeah, over to you, Ilse.
You go. Thank you very much, Francois and Maryke. That was very insightful and very thorough. And it's interesting work that you do, but I think it's always, for a researcher, it's very fulfilling to know that you are assisting someone directly, a patient. You're helping someone, and you're not just doing things in the lab without knowing the later effect. There's a question from Dr. Tumelu Satekre. It's so heartwarming to see such great outputs from research collaborations. Please expand on the causes of metabolic myopathies in the study. Did glycogen storage disease and fatty acid oxidation feature predominantly? Maryke, you're also welcome to, you can, if you click on Q&A, you can also see the question there.
Okay, I can answer that one. The metabolic myopathy study, it was specifically multiple A cell dehydrogenase deficiency. And in our case, for all the patients that we have in South Africa, they all had a deficiency in the ETFDH gene. And I think there were 5 variants, mutations, finally, of which 2 were present in all of those cases, in either compound heterozygous or homozygous. But there were 2 variants, which you can notice that in the publication that I've referenced there. But they're all in the gene ETFDH. There are 2 other MADD-like variants in the riboflavin transporter gene. One of them was previously published by the Red Cross group, and there's a new one that was also discovered later on. But for MADD itself, you know, even some of those patients included what was previously thought of as MCAD. And through this study, we could pinpoint it as a MADD, ETFDH mutation that was found in that case. And it was riboflavin treatable. So it made a huge difference in the end to have a proper diagnosis in most of these cases. So I hope that answers the question. I'm just going to check if that is properly answered from Tumelo. I cannot comment on glycogen storage disease. I don't think that they were included in the study. And the MADD were the only ones that was involved in the fatty acid oxidation from the recruitments that we had. Thank you for the question, Tumelo.
Thank you. Let me just see if there's any other questions. I don't see any other questions. You're also welcome to raise your hand. Sometimes the questions are complicated to type out. If you want to raise your hand, I can unmute you. I see in the chat, Prof. Chris Foster is saying, I associate the LMNA gene with Hodgkinson-Gilford progeria. So I assume that LMNA pathogenic variants can cause more than one disease. Can you comment?
Yes.
This is in the chat.
Yeah. So if I can refer back to my very simplified image, if you go into a deeper, more complex image, you'll see that there is so many aspects around the muscle contraction. It's not just the LMNAs, it's titin, it's I can't even remember all the other genes, but it's such a complex thing that I would assume that LMNA is not just involved in this specific gene or for this specific disease. I think definitely it can be associated with Hodgkin's-Gilford. I'm not sure what that is, though. Is it a muscular dystrophy? I'm not sure. Perhaps Prof. Izal Smuts, I'm not sure if she's in the audience, but perhaps she can also comment on this. But I haven't heard of that yet, but I know of publications associated with EMDM, LMNA specific, but yeah.
Thank you. Prof. Izal, I have muted you if you wanted to comment. If not, that's also okay. Is there any other questions or comments? I have a question that I want to ask on behalf of all the other researchers in the audience. Is there any way that a researcher can get involved in this study or can collaborate with you, or is it a closed group, or how does that work?
a closed group, Ilse, we had an opportunity to initiate this, but it's never been a closed group. There's always collaborations possible. The limitations was definitely the fellows that we had, the funding that we had, because these were the recruitment was done at academic hospitals with a massive capacity problem. So we had to get more funding and use the funding that was available for the recruitment. But I mean, at this stage, there was never actually a point that collaborations is not possible. And I know there has been collaborations already, within and outside of the group.
Okay. Is there any specific collaborations that you think can be very valuable to the groups?
Well, I think from my point of view, obviously, the data needs to be scrutinized and disseminated first properly. That needs to be completed first. But there is a myriad of variants that needs to be further investigated. There are many unknowns still of the functionality of many of these variants that was identified. The known mutations or the known pathogenic variants are easy to understand and report on, but there are many of these that we do not know. Are they indeed pathogenic? And with that, what are the functionality within the local population's genome? There's a lot of stuff that still need to be done. It definitely will keep us busy for a very long time.
Okay, and then this is a more difficult question. I see there's a lot of clinicians in the audience tonight. So the question is, should there be a physician or some clinician with a patient that they suspect has one of these defects? Is there a way to enroll them in the study or contact anyone? I know Prof. Chris Foster on the panel is also the director of the NWE program, so that might be a route as well, a different route into this. So just your opinion on that.
Answer that, yes. So the first phase of the study was the recruitment, and it ended in May of this year. So there's no more recruitment going on for the study. There's no opportunity there's no more funding left for it in any case. But I think from what we've learned, obviously, we are going ahead in any case, and opportunities like NWE is actually a fantastic opportunity to provide opportunities for where they probably wasn't before, to include to use next generation sequencing. If this is not available to clinicians, without the capacity to do it themselves, to collaborate or to bring them into the NWE project. But for the ICGNMD study, the recruitment of the phase is completed, and there is no second phase going on.
Yes, thank you very much. So I just pasted the NWE website there in the chat box. So if there's a clinician that suspect that you might have a patient and you want some assistance or input or perhaps advice, you're welcome to send a message on the NWE page. It will get to us. There's a contact form on the website. If it comes to us, I will then redirect it to either Prof. Francois or Dr. Maryke or Prof. Chris, or to all of them, and then we can all sit together and see how we can assist. Okay, I don't think there's any other questions now tonight. Maryke, Francois, thank you very much once again. This was delightful and insightful and a good way to spend a Wednesday evening. Thank you again for your time. Thank you everyone in the audience for your time. You will be contacted with regards to the CPD points that you can get for this presentation. And then I also want to thank Pfizer again. And I want to urge you, if you have any other questions, as I said, you're welcome to go to the NWE page. You're welcome to go to the National Metabolomics website and send us a message there. And I will redirect all of these questions if you think of a question later on, I will definitely redirect this to the speakers, and they will get back to you. Thank you very much. Any last words, Prof. Francois, Dr. Maryke?
To speak about this. Thank you.
Okay.
Yes, thank you.
Okay. Thank you very much. Have a good evening then, everyone.
Okay, bye.
Bye. Bye-bye.